Hi, hello. I'd like to start by thanking the organizers of the USDA's Plant Breeding Stakeholder Meeting for inviting me to give this presentation. My name is Miley Meloto, and I am a professor in the Department of Plant Sciences at the University of California, Davis. The focus of my presentation will be how plant breeding efforts can be directed to enhance food safety. I will present past successes and some future opportunities and challenges in this field. It is important to remember that almost any type of food can spread illnesses. For instance, this pie chart was created with the analysis of 1,565 outbreaks of single food commodities that occurred between 2003 and 2008. It shows the breakdown of causes of illnesses by commodity and demonstrate that raw agricultural products, including leafy greens, fruits and nuts, vine products, included tomatoes and cucumbers, have high association with outbreaks. Fresh and minimally processed produce altogether account for more than 50% of all outbreaks. The scientific community recognizes that an integrated approach is needed to deliver safer foods. All steps in the field to fork continuum can be an entry point for contamination of foods. Of concern is the contamination of crops at the pre-harvest step of this continuum, especially when a kill step is not available to clean the food. This is an area where plant breeding can and has been used to mitigate some of the problems. However, none of this would be effective without the continued effort and support for research, education, and training. With the goal of identifying knowledge gaps, innovative strategies, and research priorities in this emerging field of breeding for crop safety, UC Davis hosted a NIFA workshop in 2019. This event was organized by Dr. Alan Van Dyes, a plant breeder, Dr. Michelle J. Russell, a food safety scientist, and myself, who connects these fields of research through the study of plant microbe interactions. The workshop gathered 65 participants who represented plant scientists, breeders, extension specialists, food safety experts, and a few students and postdocs. All of these participants came from industry or government or the academia. Throughout our discussions, there was an overwhelming support for advancing research towards breeding crops with the goal of increasing the safety of food chain. We have identified the main food safety issues that include mycotoxins, the human pathogenic microbes, some heavy metals, nitrates, and allergens. However, there are some challenges that need to be addressed as well. One of the challenges identified was that we need to develop more effective assays and approaches for choosing breeding targets, and also understanding how the interaction between the plant genotype, environment, and microbe plays a role. During the workshop, we heard about several success stories, and I would like to highlight two projects where significant progress has been made, aiming at decreasing mycotoxins accumulation in grains. So one of the projects described the fusarium head blight of wheat, also known as the scab of wheat, and the other project described the Aspergillus flavus contamination of maize. Since 1998, the U.S. Wheat and Barley Scab Initiative has focused on the control of this disease. The main conclusion that they reach is that an integrated approach is required to do so. However, there are main strategies that most contribute to the management of the disease. One is the fungicide application, and the other one is the use of resistant varieties. Resistant cultivars can serve as a means of protecting wheat crops from fusarium head blight in a way that is both environmentally and economically sound. The resistance to wheat scab is quantitative, and several QTLs have been identified. The best one was named FHB1, which can reduce the symptoms by 20 to 25%. Due to its importance, this QTL was recently cloned in 2016, and the functional characterization of the genes residing in this QTL has been started. However, during an epidemic, even the best cultivar available still suffers some damage. So, additional breeding is still required to prove the genetic resistance. It has also been noted that resistant QTL is not effective in some of the genetic backgrounds tested. This might be due to the presence of inhibitors of resistance in these genetic backgrounds. Nonetheless, breeding already contributes to reduction of the disease in the field. Contamination of seed crops with Aspergillus flavus is another area where crop breeding has had some impact. Aspergillus is an opportunistic pathogen that infects the kernel when the environmental conditions are favorable. In doing so, this fungus produces aflatoxin that can accumulate in the seed and persist throughout the production chain. Fortunately, at least for maize, genetic resistance against Aspergillus growth and aflatoxin accumulation has been identified. Again, this resistance is quantitative, and the best QTL controls about 20% of the phenotype. The QTLs identified seems to be robust across different environments and genetic backgrounds. During the NIFA workshop last year, Dr. Marilyn Warburton reported that by stacking QTLs from resistant cultivars into a susceptible genetic background, they were able to reduce aflatoxin accumulation to the level of the resistant parent. In this graph, you can notice the accumulation of aflatoxin in different near isogenic line carrying individual QTLs or a combination of them. And here in the last line, where the susceptible genotype carried four different QTLs, the amount of aflatoxin accumulation was very similar to the donor parent. So this data clearly shows that these QTLs are effective and could be employed in breeding programs to control the accumulation of aflatoxin in maize. Now I am going to tell you how I envision breeding efforts to improve the safety of freshly consumed produce with a focus on leafy greens. One of the main problems concerning the safety of fresh produce is the lack of effective technologies that kill human pathogens while maintaining the quality of the crop. A student in my lab, Christian Jacob, has analyzed the severity of outbreaks that happened between 1998 and 2017 using data available at the National Outbreak Reporting System, NORS, that is maintained by the CDC. The letters O, I, H, and D below each commodity name indicates the number of outbreaks, illnesses, hospitalizations, and death episodes of human diseases caused by the consumption of fresh produce contaminated with different etiological agents listed here on the right. Hierarchical clustering revealed that lettuce and fresh tomato contaminated with Salmonella, norovirus, and E. coli are the riskiest commodity hazard pairs. Lettuce is of particular importance to the California agro-industry, and that has been the motivation for us to start this project. Tomatoes contaminated with Salmonella is more relevant to the East Coast and should also be the focus of further studies. Also, it is important to note that lettuce associated outbreaks have occurred across the entire US, and many of them originated in California fields. This is at the first steps of the farm to fork chain. This chart here does not include the three Romaine 0157H7 outbreaks that occurred in 2018 2018 that resulted in more than 374 illnesses, hospitalizations, and 5 deaths. As these human disease outbreaks are linked to crop contamination on the farm, it really begs the question as to whether plant breeding can be used to enhance fresh produce safety. Once contaminated, there's very little we can do to clean fresh produce without compromising its quality. Also, when I was talking to Dr. Trevor Soslow at the UC Davis Extension, he mentioned that after 6 years of extensive research, they concluded that contaminated fields are really impossible to clear. So, can we then capitalize on plant genetics to protect the crop against human pathogens? So, we decided to take this new approach to help mitigate the problem. So I reasoned that these human pathogens need to be somewhat adapted to survive in the plant environment and start exploring the possibility to use breeding strategies to disrupt bacterial colonization of edible leaves. We identified a few knowledge gaps and designed a study guided by two questions. One is there a variation in bacterial colonization in different lettuces? And what are the genetics and molecular mechanisms underlying this trait? To address these questions, we used a set of 11 lettuce genotypes with contrasting morphologies, color, and textures to find traits associated with decreased colonization and also traits that are robust enough to be mapped. These genotypes are parental lines of the mapping populations from the UC Davis lettuce breeding program, which can facilitate the mapping of useful traits. Dr. Richard Mitchell Moore has kindly provided seeds and had also useful discussions for this project. I thought that an important trait for genetic mapping purpose would be bacterial persistence in leaf apoplast. Internalized bacteria can be protected from routine sanitation treatments and can potentially persist throughout the food production chain and reach the consumers. Thus, bacterial persistence inside the leaf represents a relatively high risk for food safety. We calculate bacterial net growth in leaves of all 11 lettuce genotypes as a ratio of the bacterial population size at 10 days post inoculation over that of the day of inoculation, that is day 0. This analysis revealed that bacteria grew in some genotypes represented by ratio greater than 1, whereas their population size did not change or even decrease in other genotypes indicated by ratio close to 1 or lower than 1, respectively. We observed a gradient of ratios among the 11 lettuces and identified red tide and Lolo Rosa at the extremes of the gradient for further analysis. These genotypes were also chosen because they already have a mapping population derived from their crosses. We then assessed the deposition of callus on the plant cell wall as a marker of plant immune response upon bacterial infection. So, callus deposits are illustrated by the bright blue spots on the micrograph on the left side, and the area occupied by the callus deposits were quantified to create the graph on the right. Both bacteria, Salmonella and E. coli, induced deposition of callus in Lola rosa leaves, illustrated by the orange bars, but not as much in red tide leaves, illustrated by the blue bars. So these findings indicate that the ability of the bacteria to colonize the leaf apoplast is highly associated with the strength of the plant immune responses. Plant immune responses against microbes and pests are highly complex. The cartoon on the left shows key components of the plant immune system that starts with the recognition of the pathogen, followed by signaling pathway and reprogramming of the plant's transcriptional machinery. There's growing evidence gathered by several research teams indicated that various leaves can recognize Salmonella and pathogenic E. coli and initiate immune responses. One such example is also the burst of reactive oxygen species. So, to continue exploring how lettuce can mount defenses against human pathogens, we performed a transcriptomic analysis of infected leaves. After identifying lettuce genes that are differentially expressed at 4 and 24 hours post inoculation with either E. coli 157H7 on the left or Salmonella enterica on the right, we constructed the metabolic map of genes linked to biotic stress response. So each of these four maps include recognition of the pathogen, signaling, and possible downstream responses. Without going into too much detail, it is clear that both bacteria modulate lettuce stress response. However, there are some differences. First, Salmonella on the right induces changes at greater extent at both time points, indicated by the large number of genes that are differentially expressed after exposure to this bacterium. Each colored little square indicates the expression of one gene in this pathway. Second, Salmonella mostly induces plant immune responses when inside the leaf, indicated by the green squares. On the other hand, E. coli 157H7 mostly represses the genes associated with immune responses, indicated by the red square. It is clear that these two pathogens induce distinct response in lettuce, which are worth of further investigation with the ultimate goal of reducing their survival in edible leaves. The results I have thus far suggest that we can use a mapping population derived from Seriola 2 and Grand Rapids to identify genes and proteins that can control bacterial internalization rate into lettuce leaves. I have not shown this data in this presentation. But similarly, we can use the progeny of red tide and Lola rosa to identify the genes and proteins that control bacterial apoplastic persistence in lettuce. So, based on the transcriptomic analysis, I anticipated that these traits are multigenic, and the challenge would be to determine whether identified QTLs are biologically useful. To answer these questions, we need to continue foundational research to better understand the association between metabolic pathways with desirable traits and phenotypes, and how the surrounding environment and external factors such as microbes, management practices, climate can affect the metabolism and therefore affect the phenotype. If you are interested in learning a little bit more about this topic, I invite you to read a collection of articles under the research topic Breeding Crops for Enhanced Food Safety that was recently published by Frontiers. With Wei Zhan and Max Teplitsky, I have co edited this research topic, which has attracted the attention of researchers around the globe. We hope that this collection will incite new ideas and research in this field. There's still time to contribute manuscript to this collection if you are interested. I'd like to end this presentation by acknowledging students and research associates in my lab for their endless hours working in this project. Also, I thank funding agencies, several of them, especially USDA, for making this research possible. In particular, I would like to thank the participants of the NIFA workshop for their useful discussions and the insights into the future of this field of research. I thank you so much for your attention, and I'll be happy to answer any questions that you might have regarding these projects. Please email me with questions if you have them. I wish you all a productive meeting. Goodbye.